everyone. My name is Gino, Graziano. I'm a chemistry major from San Marcos City College, as was previously mentioned. My project for this summer was drug delivery with temperature-sensitive liposomes. And I was working under Dr. Joseph Zasadzynski in the chemical engineering department, and my mentor, Sally Forbes, was in the back. And the majority of our funding comes from the National Institutes of Health. So what are liposomes? They are essentially spherical objects composed of a phospholipid bilayer, which is the same material that makes up the cell membrane. And these structures are formed in aqueous solutions, so in water. And this is because the molecules themselves, the phospholipids, have a water-compatible head and a water-incompatible tail, so they kind of arrange in this fashion. And the distinguishing characteristics that make these conducive for delivering drugs are that they naturally aggregate in this leaky cancer tissue, so the vasculature is not well formed, so these large particles can aggregate. And also the ones that we're dealing with in particular are temperature sensitive, so they release whatever's inside this little bubble at around 40 degrees C, which is slightly above human body temperature. So the mechanism is that you have a drug and it permeates the membrane from some exterior force, which I'll talk about later. So the big picture is more effective cancer treatments by using these liposomes to deliver, as vehicles to deliver chemotherapeutic drugs. So here's the illustration of the tumor inside a person. So current chemotherapy, the drug, when it's administered, is dispersed in the body, which causes adverse effects on other bodily systems, so you can't give people as large dosages because it has these effects such as cell death and can also cause secondary cancers and hair loss and such. So with liposomal delivery, it's a more targeted process, so you don't have as much dispersion throughout the body, which means you can give the tumor specifically larger doses in comparison with current. And also, so this reduces the adverse effects and also allows for controlled release, which is part of my project. So research goals for this semester, or this summer, is one of the goals is what loading conditions give the highest encapsulation efficiency of the drug within these bubbles. So it's essentially looking at the amount of drug encapsulated in the liposomes compared to the total amount that you add to them. Gives you encapsulation efficiency. So here's the illustration of like a cross-section of the bilayer. So the idea is you create a difference in pH, so acidity and basicity from on the sides, the interior and exterior of the bubble. And this is kind of the driving force that pushes the drug from the outside to the inside. So while looking at these conditions to improve encapsulation efficiency, the variables I'm looking at are how much the acidity and basicity is different from the inside and outside of the liposome, and then also the temperature at which it's loaded, how long it's loaded for, and then the concentrations of both liposomes and the drug that's added to it. So part 2 of the project is using gold nanoshells for controlled release from this liposome. So the mechanism for this is that first these nanoshells here, which are just hollow gold, essentially spheres, are tethered to the liposomes with polymers, and then they're irradiated with near-infrared laser, which selectively heats the nanoshells, and then the liposome is heated by the nanoshells to its transition temperature, which I mentioned earlier was about 40 degrees C. So at that point, the membrane becomes very permeable and the drug is released. And so one of the methods that I was using to determine concentrations of the drug in solution was fluorescence spectroscopy, which is essentially you have one color of light going into the sample, that color of light interacts with the sample, and a different color is emitted, and by measuring the intensity of the color that's emitted, you can determine what the concentration is. So intensity is directly related to concentration. So fluorescence is measured before and after I essentially load the sample, then measure the intensity, then destroy all the liposomes, and then measure the intensity again. And from that change, you can see how much was inside. So there's a couple challenges to this. The photodecomposition can happen, which is where the light actually can destroy some of the drug. Self-quenching, which is essentially when the drug uses its energy that it gets from light in other ways and doesn't release as much light. Chemical interaction with other species inside the solution. So the preliminary results I got from using these methods are, so I kept everything the same except just adjusted the difference in pH from the outside, the interior and exterior of the membrane. And you can see here that there's a trend on this axis is the change in intensity before and after rupturing them. So it's indicative of how much was encapsulated. So here there's an increase of 1 as far as the difference between the acidity and basicity of across the membrane. And you can see that there's an increase in the encapsulation. So, and then also, I kept everything else the same, and all these situations have changed one variable at a time. So here is increased loading time from 2 hours to 16.5 hours, so essentially overnight. And you can see a marked increase of encapsulation. Then there is increased drug amount. So this is a constant concentration of drugs that's added. So I just increased the volume of drug that was added to a sample that contained the same amount of liposomes. And you can see an increase in the encapsulation as well. And then increased loading temperature. So from 20 degrees C, which is roughly room temperature, to 35, you can see these are 2 different comparative samples. So there's an increase in both of them as temperature was increased. So also, I looked at 2 different methods of encapsulation. The first would be a double buffer method, which is essentially when you use 2 distinct pH solutions to establish the difference in pH across the membrane. So essentially encapsulate a low pH solution and then put a high pH on the outside. The second was an ion gradient method, which essentially involves very little buffer solutions that are at a constant pH. You just add like a salt to the inside, and essentially the difference in pH is created passively. You don't actively establish it. So this showed to be much more efficient. Those ones in orange are the ion gradient, and the blue are the double buffer. So in summary of this data, the pH, the encapsulation is increased when the difference in pH is increased. Also increased when loading time is increased, when loading temperature is increased, and when the drug to lipid ratio is increased. So the ion gradient shows to be much more efficient. So as far as release with these nanoshells, as I mentioned earlier, so we loaded these liposomes using our most efficient loading scheme that we came up with, and then we have a sample that has no nanoshells in it, so just the loaded liposomes. And we first measure the intensity before shooting with the laser, then after shooting it with the laser, then after breaking it open and seeing what was inside. So here you can see there's not much of a difference in the intensity before and after radiation, which indicates that not much was released when you shoot it with the laser. So with nanoshells, before radiation, it's pretty low, and then after shooting with the laser, you see a marked increase in the intensity, which is indicative of drug being released. So, and then after rupturing any remaining liposomes, you see a small increase. So this is about 78.71% release. And during this, the solution itself was kept at about 37 degrees C, which, as I mentioned earlier, is about human body temperature. And the release temperature is also 40 degrees C, as I mentioned earlier. So any release wasn't due to the temperature of the solution, just due to the lasering. So in conclusion, increasing encapsulation is seen with increased loading time, temperature, pH gradient, and increased drug-lipid ratio. When all those increase, you see increases. So the ion gradient loading appears to be much more effective at getting the drug into the sample. And then also significant releases observed with nanoshells, with the nanoshell sample, and this suggests that there's pretty effective tethering going on between the nanoshells and the liposomes. So things that we're probably going to do in the future are leakage measurement, so how much drug naturally just leaks out of these little membranes, and then comparative release with different tethering methods. So there's more than one way essentially of getting these nanoshells attached to this bubble, and then more encapsulation data, specifically time and concentration, because we didn't have a lot of data in those 2 areas. So we did have. This is just a little 3D structure of the drug that I was working with. So I'd like to acknowledge and thank the Tally Forbes, my mentor, and Dr. Joseph Zancinski, the PI that I was working under, and the rest of the Zancinski lab for their help and assistance. And I'd like to also thank INSET for the opportunity to come here over the summer, and specifically Dr. Jens, Dr. Nick, and Dr. Erica